Good evening, everyone. My name is Larissa Moniz, and I am the Director of Research and Mission Programs at Fighting Blindness Canada, and I'm going to be your host for this evening. Before we start, I do want to acknowledge that in Toronto, which is where I'm located, we are on the traditional territory of the Haudenosaunee and the Mississaugas of the Credit First Nation. This land is also part of the Dish with One Spoon territory, a treaty between the Haudenosaunee Confederacy, the Anishinaabeg, and allied nations. And I know that we have guests and community members joining us from across Canada. And so we acknowledge the ancestral and unceded territory of all the Inuit, Métis, and First Nations people from whom this land is home. So I want to give you a very big welcome and thank you for joining us tonight for episode 1 of Fighting Blindness Canada's Accelerate Vision Health series. I want to thank our event partners because this web series really wouldn't be possible without their support. Our Ask and Answer partners, Dealey Group and Roche Canada, our virtual broadcast partners, Amgen Canada, Bayer, Heathbridge Capital Management Limited, and our registration partner, Bachner Eye Institute. So tonight we're going to be joined by two very special guests to discuss what inspires them in their careers and more broadly in vision research. There's going to be a chance for audience questions at the end of the program. So as we're having a discussion, and if you have questions that come to mind, please submit them either through the Q&A button at the bottom of the screen, or you can email us at info@fightingblindness.ca. But before we get to our discussion and I introduce our guests, I want to share a very special word from Fighting Blindness Canada's President and CEO, Jennifer Jones.
I'm Jennifer Jones, President and CEO of Fighting Blindness Canada. I'm delighted to welcome you to the Accelerate Vision Health series. There are 3 things we want you to take away from this series. First, there are research breakthroughs aimed at developing new treatments, stopping vision loss, restoring sight, and ultimately finding cures for blinding eye diseases. Second, we want to update you on the current state of vision science and the impact that mentorship has in advancing discoveries. And third, we're excited to highlight innovation in the eye health space with a glimpse into future advancements. It's important to recognize that Fighting Blindness Canada is funded entirely by philanthropy. The Accelerate Vision Health series is made possible only because of the generosity of our donors. Thank you for helping us bring the science to our community and build connections that inform, engage, and empower those affected by vision loss. This year marks our 50th anniversary, and we've celebrated the sight-saving progress made possible by your support. As some of you know, we started in 1974 as the Retinitis Pigmentosa Foundation, and thanks to you, we've expanded our scope to become the largest charitable funder of vision research in Canada. Over these 50 years, we've invested more than $40 million in research, gained insights into vision loss, and developed new treatments and cures. Additionally, together we've dedicated more than $5 million to educational programs like today's event. I'm excited that our new 5-year strategic plan, Raising Our Sights, Vision 2030, reflects our continued commitment to accelerate breakthroughs in vision research, provide expert information, advocacy, and connection for everyone impacted by blinding eye disease. Thank you for joining us today. I'm delighted to share the latest in transformational research and the future possibilities for those living with vision loss.
Well, thank you very much, Jennifer. And as Jennifer has shared, tonight's theme is really about looking and celebrating the successes of the past, particularly the last 50 years of Fighting Blindness Canada, but also looking ahead to the future. And I think this is really well illustrated with our guest tonight. So I want to welcome to the program 2 long-time friends of Fighting Blindness Canada, Dr. Ian MacDonald and Dr. Matthew Benson. Good evening to you both.
Good evening, Larissa.
Yeah, good evening, Larissa.
So I'm going to give a really quick introduction so our audience can get to know you a little bit, and then we'll dive into a discussion. So we have Dr. Ian MacDonald, who is the newly appointed head of the Department of Ophthalmology at the University of Montreal. And he recently moved from the University of Alberta, where he was past chair of the Department of Ophthalmology and Vision Sciences. And Dr. MacDonald is an internationally renowned expert on ocular genetics and with a specific focus on choroideremia. And in fact, one of his first grants was from Fighting Blindness Canada, which was named something different at that time. And also joining us, we're really happy to have Dr. Matthew Benson, who is a clinician-scientist in the Department of Ophthalmology and Visual Sciences at the University of Alberta. He holds the Bayer Professorship in Translational Research in Ophthalmology. He completed his graduate studies and residency at the University of Alberta, and during that time worked alongside Dr. MacDonald. He then completed a fellowship training in ocular genetics at the National Eye Institute. So happy to have you both here, and let's dive into our discussion. So I'm going to start with you, Dr. MacDonald. When you started your career, so sort of, I guess, looking to the past and celebrating that past now, I was wondering if you could tell us a little bit about what was the impetus or inspiration for you to pursue ophthalmology in general, but also in particular ocular genetics, which I'm assuming at the time there wasn't very much known about inherited retinal diseases.
Well, thanks for asking, Larissa, and I'll try to be fairly short. So, yes, you're right. The very first grant that I received was from the RP Eye Research Foundation of Canada. And I think that was 1984. And so that's 40 years ago. And I was then doing a fellowship in medical genetics at the Children's Hospital of Eastern Ontario in Ottawa. And my mentor at the time, Alistair Hunter, wanted me to get involved in mapping studies of human disease and molecular genetics. He thought this was the future. And so by chance, someone called from the Ottawa Citizen, and she said that in her family there was a genetic eye disease called choroideremia, and was anybody doing any research on it? And I said, well, no, not that I know. So, but then I said, well, would you like to get started? And so, sure. And so then we applied to the RPI Research Foundation for a grant. And the grant was, I think, $12,000. And boom, we were started. And off I was, seeing patients in their homes and using a handheld ophthalmoscope. And then after a while, I recognized I I need to know a little bit more about ophthalmology and inherited eye diseases, and so that's where I got started. And the RP Research Foundation, Fighting Blindness Canada, was very much a part of that start. And thank you very much.
I think it's just wonderful also, just almost by happenstance, like a conversation that led you to this wonderful career.
Hmm.
Did you ever sort of later on touch base with this same family or this person?
Well, I thought I'd be short, but in my travels to get the blood, to do the mapping studies to find the gene, I went all the way out to Alberta and drew blood on somebody who was then involved in a clinical trial of gene therapy that we started in 2015 for choroideremia. So families, um, it's all about families.
Hmm.
How about that?
Absolutely. And just a quick follow-up, Ian, to that, or it doesn't have to be quick, actually. You can take as much time as you want.
Oh,
Um, Sort of like looking back, so you started 40 years ago. I guess I wonder when you started working in this field, if you could imagine where we'd be today with the first gene therapy approved, a number of different clinical trials. So I was wondering if you could maybe tell me about what has been maybe most gratifying to you in the progress you're seeing, or if there's any big surprises in the trajectory of vision research.
so I have to think about that, but I'm going to pick something out. So I never thought that I would be involved in a gene therapy trial um, in the mid 80s. It was possible, but it would be the equivalent of a moonshot for me.
Mhm.
Um, so things have progressed dramatically where our technologies for doing genetic analysis have improved significantly, such that we can have a turnaround time of in a commercial laboratory less than a month, for example. Things that would have taken maybe years, really, well, with a good technologist, wouldn't take very long. But I'm a little bit off topic. So the rapidity of clinical diagnosis has improved significantly. And with that, we've been able to tell patients and families, this is the actual genetic disorder that you have. This is your genetic analysis. And this has enabled some of them to be linked to clinical trials. So about a year ago, the news of Luxturna became quite prominent. And there were two sisters who were in Alberta from a very small town, and they had initially been given a diagnosis of Leber's congenital amaurosis a long time ago, but they had put their names in the Foundation Fighting Blindness registry of patients to link to future clinical trials, and they came forward to actually have their genetic analysis done so that they might be enabled. Well, and so they didn't have the RPE 65 form of Leber's congenital amaurosis, and so they weren't eligible for Luxturna. But we live and work in a very small community of geneticists in North America. And one of our colleagues in Portland phoned and asked me, Ian, do you have any patients with a particular different variant in a disorder that would cause Leber's congenital amaurosis? And both of these ladies had this variant. And so I talked to them and would you be interested in, and yes, they said they lived with the hope that something might come along. So when I was talking to them, the older sister was 86 and the younger sister was in her late 70s. And so one of them did go to Portland and did have a gene therapy. And I never thought that would happen. So I hope that gives you a sense of how how dramatic that is, really, and how all of us, patients, families, physicians, are hopeful for the future. And a little story like that tells you a lot, I hope that.
No, it absolutely does. And I think it's also wonderful because I know a lot of community members sometimes maybe worry that as they're getting older, maybe as their vision loss is progressing, that they're losing the chance to be part of clinical trials or to get treatments. And I know everybody's vision, everybody's disease is different, but it is nice to hear stories like this as well, of these two sisters and of other people who have been able to access clinical trials and access a little bit of hope.
Yeah, later in life,
Yeah,
as in, yeah, just because you're over 65 doesn't mean you're not eligible.
absolutely. Yeah. I was reflecting on when you were talking about sort of the trajectory of your career. For me, I started my PhD in 2002, which was when they were just starting to, I think the first sequence of the human genome was published in 2002, or just after that. And seeing how far we have moved now with gene therapies and CRISPR and all these things. Yeah, even in sort of the time period I was doing my PhD and postdoc, it just was just so fast. Couldn't even keep up with some of the advances. Okay, Dr. Benson, I want to turn to you now. I was going to sort of ask a similar question. When you were deciding on your choices when you were finished sort of medical school and residency, what drew you to ophthalmology first, but then to ocular genetics more specifically?
Oh, yes. Thanks for the question, Larissa. Yeah, for me, like in my early years of medical school, I was kind of interested in learning about the eye and vision. And I thought it was kind of fascinating how all of these processes where light has to be first detected and sensed by the eye and then processed by the brain into something so complex like vision all occurs. It's all orchestrated by, things on the molecular level, cellular level, and then has such a profound impact on, our day-to-day life. really drew me in from kind of my early exposure during medical school. And so I explored some electives and some shadowing opportunities during my training. And I had an opportunity to see ophthalmologists such as Dr. MacDonald. I primarily worked more with Dr. MacDonald as a resident, but even as a medical student, seeing how an ophthalmologist could provide a diagnosis, management, both medical and surgical, to improve a patient's vision was something that kind of solidified my decision to pursue ophthalmology. And then in terms of inherited retinal diseases and ocular genetics in general, I'll first mention before I forget that my first grant also is through Fighting Blindness Canada. So in many ways, I'm starting to parallel some of Dr. MacDonald's accomplishments, but I can only dream to kind of get to where he's gotten. But when I first started residency, I wasn't sure I was going to go down this route. But I've always been very interested in research and kind of interested in obtaining a graduate degree at some point as well. And so I kind of had always had that interest, and I was a research representative during medical school too. But my initial goal was to become a comprehensive ophthalmologist and kind of treat patients that have a wide range of conditions. But it was very fortuitous that Dr. MacDonald has such a presence in Edmonton, and during my residency, I had an opportunity to work with him and see how his role as a clinician scientist and how he can combine both basic science research, clinical trial activities, and his clinical practice to really manage patients in a way where they can understand kind of what's going on in terms of research and what can we look forward to in the future was kind of a natural it was very natural for me to kind of pursue something similar. And so that was really what kind of drew me towards ocular genetics. And also kind of I'm coming in at a stage where we've relied on a lot of experience from individuals like Dr. MacDonald, who've kind of pioneered ocular genetics really and got us to a stage where we're already providing genetic diagnoses for patients almost routinely when they come in to see us in the clinic. And it's easy to take that for granted because, as you said, in 2002, 2003, the human genome was sequenced, and now we're delivering molecular diagnoses to patients kind of on a daily basis. So it's, there's been so much progress over such a short time period. And so I'm very optimistic for the future. But at the same time, like in clinic, as a resident and then also in practice, I'll see patients that, are really in desperate need of treatments. And so that kind of exposure as a resident, seeing that, they're patients who we really need to kind of advocate for and develop treatments through research and both clinical and basic science, kind of really drew me towards this path. And I ended up pursuing a fellowship at the States and had an excellent experience there, and was fortunate enough to come back to Edmonton to kind of share my expertise with the community here.
Thank you. Thank you so much. That's such a wonderful story and description of what has sort of inspired you to take the path you're taking. I was wondering, you've already sort of mentioned this, working with Dr. MacDonald and then going to the NEI to do some more training. I was wondering if you could sort of maybe reflect a little bit on what you've learned from some of your mentors, both in terms of being a researcher, but also in terms of being a clinician. Like, what are you taking from some of your mentors as you set up your own independent career now?
Oh, yeah, no, it's a very good question. And to touch on a few of those points, Dr. MacDonald has been my main mentor throughout residency, and continues to be so as well. He's joined me for surgical cases, even kind of aside from ocular genetics, and helped me out in many ways. And so it's hard to answer that question and give it, all of the value that it deserves. But from a research standpoint, Dr. MacDonald has opened so many doors for me. He's encouraged me to pursue this path and opened up professional opportunities that have allowed me to make collaborations. He set the groundwork for our vision science research essentially within the ophthalmology department, and the lab that I'm in currently has most of the equipment and infrastructure that he's established. And so without that, it would be very difficult for me to start up on my own, especially having clinical duties as well. And then from a clinical perspective, I was fortunate enough to shadow Dr. MacDonald during my residency for a whole year in his ocular genetics clinic. And so I kind of was able to gain all, some aspects of, the way he approaches patients, how he interacts with patients, and gain some kind of tidbits of all of his wisdom that you don't necessarily get from a textbook. And so that for me was extremely valuable. And then I kind of had that similar experience during fellowship as well, where I had an opportunity to work with a handful of kind of the world experts as well in various areas of ocular genetics. So kind of metabolic eye diseases, retinitis pigmentosa, Stargardt disease. And it was a fantastic experience where you could see patients referred to this kind of very large academic center from all parts of the US and Canada and even other parts of the world. And it was I realized that kind of I found my people when I was in those clinics because you could really sit down and talk to patients for a long time, really try to understand what's going on, answer their questions when you don't always have that luxury when you're in a busy clinical practice. And so that was something that I really enjoyed, and I hope to make time for my patients that way too, as I'm going forward in my practice now.
Thank you. I actually, we're reflecting on both what you said and what Dr. MacDonald has said. I think it makes it really clear also what a sort of global enterprise research and clinical research really is, which is interesting. We often get questions from community members about when you're funding researchers, are they talking to other researchers about the work they're doing? So, or are people just maybe duplicating results because they're not talking to each other? And I think this is a great example of, it's wonderful to see you being able to go to the States and get training, but then coming back to Canada to take what you've learned. And Dr. MacDonald, you've talked about patients going to the States, but also you moving across Canada, really that, the big spread of expertise moving around and making things stronger. My next sort of question for actually both Dr. MacDonald and Dr. Benson, we've talked a little bit about what inspired you to get into ophthalmology, to get into ocular genetics, but now looking ahead, I was wondering if you could reflect on some of the areas in vision research that you're really excited about. It could be in inherited retinal diseases specifically, it could be more broadly in ophthalmology, just some trends and future innovations you're looking forward to seeing. Dr. MacDonald, I'll start with you.
Well, I was hoping you would start with Matt because he's working on some wonderful things. He's working on little subunits of the cell called peroxisomes, which is so exciting. So anyway, okay, so I'll go back. Uh, um, I'm very excited of the possibility of gene therapies that are not only replacing genes, but actually repairing genes. And so in a nutshell, I think that's really exciting. We've heard about CRISPR-Cas9, but there are other mechanisms that one can use to repair and prevent an abnormal protein, for example, from being formed, and especially in the eye. And so they're not all going to work, but some of them are going to work, and some of them have worked. And we're still in that phase of experiment, but I do see this as being very hopeful that there are therapies that are coming along, not in 10 years, but in a much shorter period of time, perhaps in the next 5 years. And there are things in the so-called pipeline, and industry is involved. Before, this was very much uh, an enterprise that you did because you were interested. But we need industry to be involved, and industry is very much involved, and that's very nice to see. So I'll stop there.
I just want to follow up when you mentioned, and don't know if it was a specific comment or more of a broad comment. You said something about especially in the eye, some of these things like gene editing can happen. Is there something specific about the eye that makes it more likely that some of these therapies are coming in the eye?
Well, one of the concepts that we must consider is that there are so many genes that are functioning in the eye. And so there's a wealth of opportunity there. There's so many eye conditions for which we know what we know the genetic cause is. And now we're trying to figure out how to treat that, and not treating the whole body, but just treating the eye. There are some risks, obviously, with treatment of the eye, but still, it's always been a fascination that perhaps we can do this in the eye where we can't do it elsewhere.
Okay, thank you. And Dr. Benson, a similar question for you. What are some things as you're now at the start of your career, looking forward in 10 years, 15 years, 20 years, where do you think where do you think we might be?
Hmm.
Yeah, no, thanks, Larissa. I'd echo what Dr. MacDonald says and add that for the last, 10, 20 years, we've heard about precision medicine. And I think a lot of that at the moment has been around diagnostics. And like we were mentioning, already we're able to provide specific molecular answers for patients, explanations for patients' vision loss that we weren't able to do 10, 20 years ago, or at least not routinely. So there's been all of those advances already at this point, and now I think that paves the way for specific precision medicine treatments. And so, like Dr. MacDonald mentioned, I think that's, it's not as far off as we think. And in terms of like a lot of the focus is on inherited retinal diseases, of course, and that's where the first gene therapy that's Health Canada approved came from. But there's also a lot of precision medicine approaches to other more common disorders like macular degeneration and glaucoma. I'm not necessarily an expert in some of those aspects, but even some of the experiences that we've learned from genetic therapies, such as in Stargardt disease, for example, through clinical trials, are applied to conditions like macular degeneration and trying to slow the progression of geographic atrophy. And so even though a trial maybe is negative, is of course disappointing for us and for patients, but we learn so much from them. And a lot of what we learn can be applied to the treatment of other ocular conditions too. And so I think that it almost seems like a dam waiting to break open where, we've had already some successes, but there's lots of research going on in the background to understand the conditions and then also to develop treatments. And so I'm very optimistic for what's to come for patients.
I think you've sort of alluded to the idea that there are sometimes some successes, but there are also some failures. And I guess that's the nature of research, is you ask a lot of questions, and sometimes the answer is what you want to see, and sometimes you get more information, but maybe not quite what you were hoping for, and then you have to try a different way.
Exactly. And sometimes you get a sense like in certain patients, perhaps the clinical trials seem to be beneficial, but when they kind of averaged everybody, it maybe didn't. And it also speaks to clinical trial design and trying to develop kind of outcome measures that are going to be sensitive to detect whether or not the treatment is successful. So we're learning about clinical trials as well as learning about the conditions through research.
I think that's like another really important point is so many of these sort of innovative therapies, whether it's for an IRD or whether it's for a stem cell therapy for AMD, they are so innovative that we don't necessarily know what the endpoints are that we need to look for yet. And so there is just designing a clinical trial that is going to show if a treatment is safe and effective. It's not trivial just to set up the trial itself. So we were talking about future innovations, and I think this is a great moment to segue to share a bit of, I think, exciting news. So this year, Fighting Blindness Canada, we held a research grant competition, and we really focused this year on early career investigators. And I'm really pleased to be able to share that Dr. Benson actually is one of the recipients of our 2024 Fighting Blindness Canada Research Grant Award worth $184,000. So congratulations, Dr. Benson.
Yeah, thank you so much, Larissa, and thank you, Fighting Blindness Canada. This organization has supported me for many years, and I'm very thankful to all the donors that are as eager as I am about research and where we're going to take things in the future.
Absolutely. And we actually had an opportunity to visit your hospital in Edmonton, and you discussed a little bit about what the award means to you and about the project. So we have a very short video clip that we are going to share now.
Hello, I'm Dr. Matthew Benson. I'm an ophthalmologist and clinician-scientist. I work out of the Royal Alexandra Hospital, and I also have a research laboratory at the University of Alberta. The Early Career Researcher Award from Fighting Blindness Canada is, not only does it provide funding for my lab and for us to kind of advance our vision research efforts, but also just personally, it motivates me to kind of continue along this line of work. It isn't necessarily an easy path to follow, having both clinical and research roles. And so having funding like this really, it motivates me to continue along this line of work and really push to develop therapies for patients. My research involves looking at a specific cell type within the retina. When a particular component of the cell is not functioning properly, patients lose vision. And so my lab is trying to understand why this happens. And ultimately, our research efforts are going towards developing a treatment to prevent vision loss in patients with these problems. So our plan is, once we learn more about these conditions in the lab, is to translate them to the clinic. And that usually involves developing a clinical trial to test specific treatments to see if they'll be helpful for patients. And that's what we aim to do going forward. The donors from Fighting Blindness Canada, while you don't necessarily have an opportunity to interact with the patients and see on the ground level what your funding is really doing, in the clinic I'm able to kind of work with these patients and see the translation of the research efforts that you're supporting and how much of an impact it has on the lives of Canadians. And so I thank you for all of your support along this journey. On behalf of myself, my lab, and everyone in the clinic, thank you Fighting Blindness Canada for providing this funding and opportunity to support my work.
So now we are going to open it up to the audience for questions. It is our ask and answer session, which is sponsored by the Dealey Group and Roche. And so as I mentioned at the beginning of the webinar, you can put any questions into the Q&A section at the bottom of your screen, or you can email us at info@fightingblindness.ca. So we have had a couple of questions come in already, so I will start with that. There was a question about beyond gene therapy, because we have talked a lot about gene editing and gene therapy type procedures. What progress is there for stem cell therapy? And they asked specifically for retinal pigment epithelial cell regeneration. But I don't know if either Dr. MacDonald or Dr. Benson, if you have any thoughts about this.
I'll let Matt go first.
Oh, sure. So I'd say kind of during fellowship, the kind of conversation was usually that we're at quite an experimental stage from stem cells in general. Although there are some early clinical trials where they're injecting stem cells into the eye to see if they secrete a protective factor that can be beneficial at preserving photoreceptors, for example. There have also been some clinical trials looking to deliver retinal pigment epithelial cells, which are kind of the supporting cell layer of the retina, back into the eye, which requires a surgery. But that's still at the safety stage right now. We're not sure if that's going to be something that's going to support vision going forward, but it's mainly to determine if that's going to be something safe to do. And the retina, as you can imagine, is quite a complex structure, and not only needs the support cells, but you also need the light sensitive cells like the photoreceptors and all of the integrating neurons and the retinal ganglion cells. And so it's challenging to entirely replace the retina, and that's not necessarily what you need to do to kind of restore vision, but to have kind of significant gains when you're missing cells, you need to give back sometimes more than just the supporting cells. So that's been part of the challenge with the development, but there's lots of research going on both at the fundamental science level as well as in clinical trials to try to push that a bit further.
And maybe I could say that the advent of using stem cells from the patient and then differentiating those stem cells into eye cells has been very important in the pathway of developing clinical trials. It's provided the preclinical data. So whereas let's say stem cell transplantations are possible, all the preclinical work that goes into a clinical trial has been enabled. Well, much of the work has been enabled by stem cells and differentiating those into eye cells and then treating those cells and then showing the regulatory bodies that these treatments actually work and changing the cellular dynamics.
Mm-hmm. And this year, actually, we heard about one of the first photoreceptor stem cell therapies that got approval from the FDA to start a clinical trial. And we were really excited because we have funded work through Dr. David Gam, who to support some of that sort of preclinical work, exactly like you're saying, Dr. MacDonald. Fighting Blindness Canada funding helped fund that preclinical work so it could move to a more clinical trial setting. So Dr. MacDonald, we have a question specifically for you that says, hello to Dr. MacDonald, because someone who's a former patient of yours and has seen you a number of times over the past decade and lives in BC. They were diagnosed with RP, autosomal dominant, and they have a Rho gene mutation. And so they were wondering if there were any treatments on the horizon for this particular retinitis pigmentosa. They gave the specific gene mutation, but I don't know if I won't say it out loud here, but if you can maybe talk more broadly about maybe Rho gene therapy approaches for specifically for autosomal dominant.
Right. So there are gene therapies that are designed for specific variants, and there are, so let's say specific variants in a specific gene, like in the rhodopsin gene, or there are other therapies that are just designed to really treat perhaps a whole series of patients with retinitis pigmentosa, not just those that are due to variants in the rhodopsin gene. So it really is important to know what the variant is. So I would encourage the questioner to sense, he or she knows where I live.
Yeah, they do know the variant. So yeah, maybe they can
They could reach out to me and I'll be able to give them a more specific answer.
Yeah.
So there are trials that are designed to consider using a gene editing approach for rhodopsin. Some of them are in the developmental phases, and it really would be helpful to know exactly what the variant is. So I don't want to mislead the questioner or the audience as well. And there is a clinical trials website that one can look at, and it's open to the public. It's clinicaltrials.gov, G-O-V. And you can go on that website and you can search for retinitis pigmentosa, for example, and find out what trials are ongoing, if they're nearby, and who is in charge of that trial, and what's the number to call. And so don't depend on us. You can go out there and do some searching yourself as well. And Matt and I have the great pleasure to work with a genetic counselor who is very much an enabler and a link for patients who are asking this type of question. And Matt, I would let you fill in the blanks as I started the answer.
Oh yeah, no, I don't have a whole lot to add apart from, as Dr. MacDonald was alluding to, some of the therapies don't necessarily depend on a specific variant in the gene or even the gene itself. Some can be like small molecules that potentially slow down the progression of disease, or kind of in most patients with retinitis pigmentosa. And in others, there are gene therapies that are not necessarily delivering that particular gene, but a different gene that is thought to still be helpful for patients that have changes in, say, the rhodopsin gene. So there's a clinical trial, I think it's a phase 2/3 by Ocugen, where they're delivering by subretinal gene therapy a different gene, but it's thought to potentially be helpful in patients with rhodopsin as well. So still an experiment in the clinical trial phase, but a lot of it does depend on the specific variant in the gene, but there are other approaches that don't necessarily rely on that information too.
Yeah, and I think that's a good point. We talk about them using the term gene agnostic, which I think is actually quite a confusing term sometimes. But that just means it's a therapy that isn't specific to maybe a specific gene mutation. And I know it gets complicated when you talk about putting in another gene for a different gene, but those are some of those broader non-gene specific therapies. So I've got a couple of sort of broader questions here. So the first one was saying that there's Patricia said that her son was diagnosed 10 years ago with RP. He's now 51. When he got DNA sequencing, he didn't have any of the known mutations. And he now has a level of vision that he's been told that his vision is too far progressed, or vision loss is too far progressed for any potential therapies. So I think it was sort of more of a statement, but like she was sort of asking what can they do about this in terms of potential trials or other options.
Maybe, Matt, maybe I'll start. So there are some things that might be helpful. So for example, there's a clinical trial in the United States, and I think there are some Canadian centers that are involved as well, using a nutritional approach, using a something called N-acetylcysteine. Or NAC. It's an antioxidant. We believe that light coming into the eye creates an oxidizing environment that may be quite harmful, especially in patients with an inherited retinal disorder. And anecdotally, I will say that I have given advice to patients to take this NAC, and they did perceive that there was an improvement in the visual function with it. And in fact, in one case, an individual took an acetylcysteine, and afterwards we did electrophysiology, and that improved. And then he stopped and had a period off the NAC, and his vision wasn't as good as it was, and his electrophysiology wasn't as good as it was. And then we repeated the test, and in fact, his vision got better with the antioxidant. So there are nutritional approaches that are important, even though um, even though it seems that there's nothing that can be done. I do think the nutritional approach is important. So a review of uh, what uh, what the patient is actually taking. I always talk about making sure that you have enough fish in your diet. If you have fish once a week, for example, if you don't, well then supplement your diet with docosahexaenoic acid. You can buy it at Costco or wherever, local pharmacy. Those sort of things are helpful as well. And stopping smoking. I better stop. I'll let Matt fill in some blanks.
Oh, sure. Yeah, I just might add to it. Maybe it depends on the clinician that saw your family member, whether like maybe they came at it from an approach that, well, gene therapy might not be effective because there's too much retinal degeneration that's already occurred. But
Mhm.
there are kind of different treatment approaches that target different stages of macular or retinal degeneration. And so it's possible that, in some of the early clinical trials that are ongoing right now, they're trying to deliver an agent that makes a cell type sensitive to light that's not normally sensitive to light. And so that potentially could be, applicable in patients with very advanced vision loss. But there are kind of nuances around that as well. So I guess we'd need some more information as to kind of the condition of the patient. But I think that there probably are still some ways to go about giving hope to the patient, and even something that might be effective, like what Dr. MacDonald was saying.
Thank you. We actually had a question that was specifically about research. So somebody was asking, what would you say is a primary expense for researchers, and where is funding most useful? Is it equipment, staff, students, chemicals, reagents? Matt, I'll start with you.
Oh, sure. Probably the biggest expense, but also the most important expense is personnel. And so that is something that researchers have to rely on funding in order to employ the people that really make the research happen. So I have kind of more of a supervisory role, but the people that are on the ground, actually doing the experiments are graduate students, research technicians, research associates, and their funding comes from grants that we have to apply for, and that probably takes up at least half or more than that of funding. And so it's tough because it's difficult to get funding in general, and without it, we don't have the personnel that can really do the research. So certainly equipment and reagent costs are up there too, but I would say personnel is the big investment.
Yeah,
And
for many of us, that is very much the case. We have students who very much are part of our research lab. They help us with ideas, but it takes a fair amount of time for the student to become productive. And so we need a research technician. The research technician demands a salary that's relatively competitive. And so grant funding, such as was given to Matt, is very important to keep those people in place. Training young vision scientists, training our students, doing the ordering, making sure that the equipment is running. All of those tasks require somebody who is physically present, um, and it is a challenge.
Got two, I would say, more logistical questions. One of them was about the recommended dose for N-acetylcysteine. And is that something that is a standard, or is that something that somebody should be talking to their eye doctor about?
Oh, well, you can take it over the counter. It's very safe. The side effect might be a bit of an upset stomach, but if taken with a meal, it shouldn't be a problem. So that usually I would say take 500 milligrams a day. The N-acetylcysteine or NAC trial, NAC, is 500 milligrams twice daily. So depends upon how the patient tolerates it.
Okay. And the other one is to clarify if, Dr. Benson, are you seeing Dr. MacDonald's patients now that Dr. MacDonald is in Montreal?
Yes, yeah, I have
Okay.
started seeing a number of his patients, yeah, and I'm taking on most of the patients, if not all of them. Anyone that
Okay.
would like to see me can see me.
Perfect. Um,
He's much smarter than I am. So
Oh, I don't know.
well, he could always call on Dr. MacDonald if you have any questions. So that's probably
I do
nice
it all
to
the
know.
time.
Yeah. Okay, so here's a question. I think I will start off with Dr. MacDonald for this one. It was someone who has been diagnosed with choroideremia, which has been confirmed by genetic testing. They are now 53 and they have no peripheral vision and cannot see in dim light. And they were saying that they recalled there were some clinical trials in recent years and was curious, sort of very briefly about how they went and what future trials may be in the works.
Okay, so we did a clinical trial using what is called a viral vector. So a disabled virus carrying the normal copy of the gene to replace the existing copy in the eye. And unfortunately, in one case that was treated in Edmonton, the patient had an immune reaction in the retina. So the vector is a disabled virus with a protein coat. There are resident immune cells in the retina that were triggered, and an inflammation started that was controlled to a certain extent with a high dose steroid. But once the inflammation started, then not only did it injure the retina, but it also silenced the vector itself. And so this dampened my personal enthusiasm for using this approach in the retina. The retina in choroideremia is fragile. It's detached with a subretinal injection. It's not without risk. And there were no further patients than 6 that were trialed in Edmonton. There were no other ones that were recruited into a continuation of the trial. And Biogen, one of the companies that took on the choroideremia trials globally, let's say, reported in 2021, I believe. I'm sorry, they first reported in 2021, and then later, I think 2023, that the trials were not successful. And so an alternate approach to using a subretinal injection and using a viral vector carrying a replacement gene is something that many people are thinking about, many people are working on. There was another approach that was designed to inject the vector with the gene, not underneath the retina, but into the vitreous, into the cavity and behind the lens. And anecdotally, and patients talking to me, I understood that there was significant inflammation in the vitreous. And so I'm not sure of the status of those trials, but I don't think that that as well is a very good approach because you're putting a large volume of vector into the eye, not just a small volume right underneath the retina. So more virus going into the eye, more opportunity for inflammation. But that's not the end of the story. Part of the story with the gene therapy trial in Edmonton is that we found 2 patients that had a very specific variant in the choroideremia gene that might be treated using a totally different approach, but an approach that required genetics and genetic understanding, but to use what we call an RNA, much like the RNA that is creating the antibodies for us. This is a highly modified RNA that will bind to the pre-RNA within the cells and prevent an abnormal copy of the choroideremia gene from being formed. And so there are other trials using a modified RNA that have been used for other genetic eye diseases, and there have been successes. So I'll tell you about one of those. That's the CEP290 form of Leber's congenital amaurosis. There's a product called cipofarsen. It's an RNA, a modified RNA. It's injected into the eye, and many of the patients who received that had benefit. One of the patients was from Alberta, and he was a carpenter, had a cell phone. I would use the cell phone to receive messages, but he could never read this actual cell phone. He had this therapy with an injection into the vitreous, and afterwards he could actually read the time and had really a dramatic improvement. So that's an RNA therapy that was used for a totally different disorder. But our group is still trying to see if we can develop an RNA-based therapy for choroideremia for a very specific variant that we found. We have cells from the patient. We treat the cells for the patient with the RNA, and that will enable a normal protein to be formed. So in a nutshell, probably trying to use language that is not too confusing, gene therapy using a virus didn't work very well for choroideremia, but gene editing with something like an RNA might be on the horizon. We just have to be able to take it from what we call the bench to the bedside. And that is a challenge because it takes research, the preclinical work, and it tends to be quite expensive.
Thank you so much. We have a lot more questions, which we don't have time for, unfortunately, but for anybody whose questions was not answered today, you can email our health information line or call it. The email is healthinfo@fightingblindness.ca, and we will do our best to get you the information to answer your questions. For instance, we have information about clinical trials. We can help you navigate the clinicaltrials.gov website that Dr. MacDonald talked about because it can be a little bit confusing. So please feel free to email us and we will do the best we can. So I want to thank everybody for engaging in the discussion, and specifically Dr. Benson and Dr. MacDonald for joining us. We really appreciate you sharing your time and, of course, for your commitment to helping us accelerate vision health for the community. So thank you both very much, and I will let you both go.
Oh, thanks very much, Larissa. It was a pleasure to be here. I talked too much. I'm sorry.
No, we've loved having you.
Yeah, thanks, Larissa, and thanks Fighting Blindness Canada for putting on this event.
Thanks everybody for listening too.
Okay, and so I've got a last few items. So I'm going to invite you to join us again in 2 weeks on Thursday, November 21st for the second episode of our Accelerate series, where we'll be talking about navigating eye care and the state of vision health. And I want to say another thank you to our Accelerate event sponsors, Dealey Group, Roche Canada, Amgen Canada, Bayer, Heathbridge Capital Management Limited, and the Bachner Eye Institute, as well as a thank you to our community sponsors. And for anyone out there, if you were inspired today and would like to support vision research like Dr. Benson's, you can donate using the QR code that is on the screen or at fightingblindness.ca. And we always appreciate any support that you're able to give. And finally, we're ending tonight's episode with a very special video that spotlights one of our community member stories, which I hope you'll enjoy. And at the end of the video, when you exit the session, a survey will pop up and we would love for you to share your feedback with us. So thank you very much and enjoy the video.
I may not be able to see the world as I used to, but I'm still living a life on my own terms. As an immigrant to Canada over 50 years ago, I've always been independent, but in 2020, I was driving one evening. I realized there was something wrong. My vision was fading. The diagnosis of age-related macular degeneration was devastating. My first reaction was, where do I go from here? I had to make some tough adjustments. I lost my driver's license. It felt like I was losing my independence. We felt that it might be a good time to downsize, to move from a multi-level house to a condo in the city. With the help of my wife and my family, it turned out to be a blessing. Here, I'm closer to my doctors, my community, and the support that I require. I've learned to accept my conditions and focus on what I can control. Now I'm helping to spread the awareness. The one thing that I would like others to know, don't neglect your health. Stay informed, keep up with your doctor's appointments, and don't be afraid to ask for help. Your eyes are the gateway to everything. Take care of them.